Microscope, an instrument that produces an enlarged image of an object. Microscope का use cells की study करने में, cell parts and organisms that are too small to be seen with the naked eye. Two types of microscopes. Light microscope. Light passes through one or more lenses to produce an enlarged image of a specimen. Electron microscope forms image of a specimen using a beam of electrons rather than light. Zacharias Janssen, Dutch spectacle maker who tested several lenses in a tube and discovered that nearby objects appeared significantly enlarged. Anton van Leeuwenhoek, Dutch merchant who learned how to grind lenses to make simple microscopes, have only one lens. These produced clearer and more enlarged images than Hooke's. He is considered the father of microscopy and built over 500 different microscopes. He was the first to discover microorganisms under a microscope by observing a drop of pond water filled with life. He called them tiny animalcules. He also saw and studied bacteria and the blood flow through capillaries in the tail of a fish. Robert Hooke used a microscope to observe a thin slice of cork. The spaces he saw reminded him of the small rooms where monks lived, so he called them cells, which he used to describe the smallest. He used microscopes with 2 and 3 lenses, but they didn't produce very detailed images. The electron microscope. Resolution is the limiting factor to a light microscope, since the greater the magnification is, the less it is able to resolve the image. At magnifications beyond 2000x, the image becomes blurry, but electron microscopes can be used at greater magnifications. Characteristics of the electron microscope. A beam of electrons is used to produce an enlarged image of the specimen. It does not use light. This electron beam in the specimen must be placed inside a vacuum chamber so that the electrons in the beam do not bounce off gas molecules in the air. Since living things cannot survive in a vacuum, the electron microscope cannot be used to
There are 3 major parts to the compound microscope: a body tube, an objective lens, and an eyepiece. The body tube is comprised of 2 smaller telescoping tubes with lenses coaxially mounted at either end. The wider tube holds the objective lens, while the narrow tube holds the eyepiece. The knobs for both coarse and fine adjustment allow the tubes to slide one into another, varying the distance between the 2 lenses. The lenses are convex with different focal lengths. The lens closer to the object is the objective and has a short focal length, FO, usually below 1 centimeter. The lens nearer the eye is the eyepiece. Although the eyepiece also has a short focal length, FE, it is still greater than the focal length of the objective. A revolving nosepiece, known as a turret, is attached to the lower end of the objective. The turret can be rotated to change the power magnification as necessary. Below the objective sits a flat platform known as the stage. On the surface of the stage, 2 stage clips hold the slide in place. A small aperture is present in the stage, allowing light into the body. The body tube is arranged on an arm which is connected to a strong base. This arm allows the body of the microscope to tilt. A small mirror at the base of the microscope reflects the light through the stage aperture. Above the mirror, an iris, also known as a diaphragm, is present. Iris helps in controlling the amount of light passing into the stage aperture. Working of the compound microscope. The object O is placed just outside FO, the focus of the objective lens. A real, inverted and magnified image, I1, is formed. The magnified image, I1, acts as an object for the eye lens. The eyepiece is adjusted such that the image I1 lies within the focus of the eyepiece, FE. The eyepiece functions as a magnifying glass and forms a virtual, erect, and magnified image, I2, of the object. Magnification of the compound microscope. The magnifying power of a lens is expressed in diameters. A lens magnifying an object 5 times has a magnification of 5 diameters and is written as 5x. The total magnification of a compound microscope is the product of the magnifying power of the objective and the magnifying power of the eyepiece. For example, if the objective has a magnification of 40x and that of the eyepiece is 10x, then the magnifying power of the microscope is 400x.